Hello everyone. My name is Ivy. And today I'm going to be talking about genetics, reproducing life and producing variation, created by Tone. He is my owner. First of all, I would like to introduce the topics for today. 1. The cell, its role in reproducing life and producing variation. 2. The DNA molecule, the genetic code. 3. The DNA molecule, replicating the code. 4. Mitosis, production of identical somatic cells. 5. Meiosis, production of gametes, sex cells. 6. Producing proteins, the other function of DNA. 7. Genes, structural and regulatory. 8. Polymorphisms, variations in specific genes. 9. The complexity of genetics. Let's start with the first one, the cell, its role in reproducing life and producing variation. The cell is the basic unit of life for all organisms. Every organism has at least one cell, that is the baseline definition of an organism. Organisms having cells with no internal compartments are called prokaryotes. Organisms with internal compartments separated by membranes are called eukaryotes. The membranes enclose the two main parts of individual cells, the nucleus and the cytoplasm, between which various communications and activities happen. Eukaryotes are multi-celled organisms that have a membrane-bound nucleus containing both the genetic material and specialized organelles. The nucleus is a membrane-bound structure in eukaryotic cells that contains the genetic material, and cytoplasm is the jelly-like substance inside the cell membrane that surrounds the nucleus and in which the organelles are suspended. In all animals and plants, there are two types of eukaryotic cells. The first one is gametic cells, also called body cells, compose most tissues, such as bone, muscle, skin, brain, lung, fat, and hair. The second one is gametes or the sex cells, sperm in males and ova, or eggs, in females. In humans, somatic cells are diploid, having 46 chromosomes, whereas gametes are haploid with 23 chromosomes. Diploid is a cell that has a full complement of paired chromosomes. And haploid is a cell that has a single set of unpaired chromosomes, half of the number of chromosomes as a diploid cell. The DNA molecule, the genetic code, the chemical that makes up each chromosome, DNA, is the body's genetic code. Each DNA sequence, each protein generating code, is a gene, and the complete set of genes in the genome. In addition to the identification of specific genes, the functions and interactions of many genes are becoming known in remarkable detail. Within any organism, DNA is homoplasmic, meaning is the same in each. And every cell the DNA in a skin cell matches the DNA in a bone cell. A small but significant amount of DNA is contained in tiny organelles, called mitochondria, within each cell's cytoplasm. These structures use oxygen to turn food molecules, especially sugar and fat, into adenosine triphosphate, which called ATP, a high-energy molecule that powers cells and, in turn, powers every tissue in the body. In theory, a maternal lineage, or matrilineage, can be traced back hundreds of thousands of years. Unlike in DNA, mtDNA is heteroplasmic, meaning it can differ among different parts of a person's body or even within the same kinds of cells. The DNA molecule is the blueprint of life. Understanding this structure is key to understanding the growth of any organism, and the transmission of genes from parents to offspring. On each side of the ladder, every unit of sugar, phosphate, and nitrogen base forms a single nucleotide. While the sugar and phosphate are the same throughout DNA, the base can be adenine, which we can call A, thymine is T, and guanine is G, or cytosine is C. The next one is the DNA molecule, replicating the code. One function of the DNA molecule is to replicate itself. Replication takes place in the nucleus and is part of cell division, leading to the production of new somatic cells, which call mitosis, or the production of new gametes calls meiosis. Replication thus results in continued cell production. From the single-celled zygote like the fertilized egg to 2 cells, then 4 cells, and so on. In replication, DNA makes identical copies of itself. Going from one double-stranded parent molecule of DNA to two double strands of daughter DNA. Within somatic cells, chromosomes occur in homologous, or matching, pairs. Each pair includes the father's contribution is the paternal chromosome, and the mother's contribution is the maternal chromosome. These non-sex chromosomes are called autosomes. Another one is the karyotype, or complete set of chromosomes, includes all of the autosomes and one pair of sex chromosomes. The characteristics of the chromosomes for an individual organism or a species, such as a number, size, and type. The karyotype is typically presented as a photograph of a person's chromosomes that have been arranged in homologous pairs and put in numerical order by size. Females have two X chromosomes. Males have one X and one Y chromosome. The Y chromosome can be highly informative about paternity over many generations. The matrilineal-based UnEssay, which is passed on only by females. UnEssay goes to all of a woman's children, whereas the Y chromosome is passed only to a man's son. Okay, so right now, let's watch a video on YouTube is about the step of DNA replication.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
The next one is the number 4 is mitosis, the production of identical somatic cells. A diploid cell, a cell having its organism's full set of chromosomes, divides to produce two cells, each of which also has the full set of chromosomes. There is the two steps of DNA replication. Step 1 is the strand of DNA unzips to form templates, which is weak nucleotide bonds between bases break, exposing two parental strands of DNA. And step 2 is templates plus nucleotides yield daughters, which is free-floating nucleotides in the nucleus match with the newly exposed template strands of DNA. Free-floating nucleotides are nucleotides that are present in the nucleus and are used during DNA replication and mRNA synthesis. The number 5 is meiosis, production of gametes which call sex cells. The genetic code is transmitted from parents to offspring via the female and male gametes. Gametes, remember, have only half the chromosomes that are in somatic cells. They are haploid, containing one chromosome from each pair. Each gamete contains just one chromosome from a homologous pair, and just one sex chromosome. During reproduction, each parent contributes only half of his or her genetic material. Contains your mother's chromosome or your father's chromosome is completely random. Homologous chromosomes often exchange parts when they pair up and intertwine is called crossing over. As Mendel had recognized, each physical unit, that is, gene, passes from parent to offspring independently of other physical units. This independent inheritance is often called Mendel's law of independent assortment. Law of independent assortment is Mendel's second law, which asserts that the inheritance of one trait does not affect the inheritance of other traits. There are some keywords that we need to know. Recombination is the exchange of genetic material between homologous chromosomes, resulting from a crossover event. Haplotypes are a group of alleles that tend to be inherited as a unit due to their closely spaced loci on a single chromosome. Haplogroups are a large set of haplotypes, such as the Y chromosome or mitochondrial DNA, that may be used to define a population. Translocations are rearrangements of chromosomes due to the insertion of genetic material from one chromosome to another. Non-disjunctions refers to the failure of the chromosomes to properly segregate during meiosis, creating some gametes with abnormal numbers of chromosomes. Monosomy refers to the condition in which only one of a specific pair of chromosomes is present in a cell's nucleus. And trisomy refers to the condition in which an additional chromosome exists with the homologous pair. Number 6, Part 1 is the producing proteins, the other function of DNA. DNA serves as the template for protein synthesis. Proteins are the complex chemicals that make up tissues and bring about the functions, repair, and growth of tissues. Proteins consist of amino acids, of which there are 20. Most of the human body is composed of proteins, and the body produces 12 amino acids. Amino acids are organic molecules combined in a specific sequence by the ribosomes to form a protein. Most of the human body is composed of proteins and the body produces 12 the amino acids. The other 8, also called essential amino acids, are the amino acids that cannot be synthesized in the body, they must be supplied by the diet. Structural proteins that form an organism's physical attributes are responsible for physical characteristics, such as hair form, eye color, tooth size, and basic bone shape. Regulatory also proteins that involved in the expression of control genes includes enzymes, hormones, and antibodies. Number 6, Part 2. Protein synthesis is a two-step process. The first step is transcription, takes place mostly in the cell's nucleus, and involves the creation of messenger RNA, mRNA, based on the DNA template. The second step is translation, takes place in the cytoplasm, and involves the transfer of amino acids by transfer RNA, DNA, to the ribosomes, which are then added to the protein chain. A single template for another kind of nucleic acid is ribonucleic acid is a single-stranded molecule involved in protein synthesis, consisting of a phosphate, ribose sugar, and one of 4 nitrogen bases. RNA has the same nitrogen bases as DNA, except that uracil replaces thymine and matches with adenine, while guanine continues to pair with cytosine. This is the last part for number 6. For the protein synthesis, there are also some keywords that you need to understand. First of all, Messenger RNA, mRNA, is the molecules that are responsible for making a chemical copy of a gene needed for a specific protein. That is, for the transcription phase of protein synthesis. The second one is ribosomes, is the organelles attached to the surface of the endoplasmic reticulum, located in the cytoplasm of a cell. They are the site of protein synthesis. The third one is ribosomal RNA, rRNA, is a fundamental structural component of a ribosome. The fourth one is transfer RNA, tRNA, is the molecules that are responsible for transporting amino acids to the ribosomes during protein synthesis. The next one is anticodons, is sequences of 3 nitrogen bases carried by tRNA. They match up with the complementary mRNA codons, and each designates a specific amino acid during protein synthesis. Next is triplets, are sequences of 3 nitrogen bases each in DNA, known as codons in mRNA. Next is codons, are the sequences of 3 nitrogen bases carried by mRNA that are coded to produce specific amino acids in protein synthesis. And the peptide bond is a chemical bond that joins amino acids into a protein chain. And next is polypeptide, also known as a protein, a chain of amino acids held together by multiple peptide bonds. The next one is coding DNA, is a sequence of a gene's DNA, also known as exons, that are coded to produce a specific protein and are transcribed and translated during protein synthesis. The last one is non-coding DNA, is a sequence of a gene's DNA, also known as introns, that are not coded to produce specific proteins and are excised before protein synthesis. Number 7 is genes, structural and regulatory. Structural genes are responsible for body structures, such as hair, blood, and other tissues. Regulatory genes turn other genes on and off, an essential activity in growth and development. An organism's form and the arrangement of its tissues and organs are determined by regulatory genes called homeotic, HOX, genes. These master genes guide, for example, the embryological development of all the regions of an animal's body, such as the head, trunk, and limbs. The first part of number 8 is polymorphisms, variations in specific genes. Along each chromosome, a specific gene has a specific physical location, or locus, plural, loci. Each person has one of 4 blood types A, B, AB, or O. And these 4 types compose the ABO blood group system. A genetic trait such as this one is called a polymorphism. Polymorphism refers to the presence of 2 or more alleles at a locus, and where the frequency of the alleles is greater than 1% in the population. The specific proteins, on the surface of cells, that stimulate the immune system's antibody production are called antigens. And, molecules that form as part of the primary immune response to the presence of foreign substances. They attach to the foreign antigens are called antibodies. And the next one is Mendel's first law, which asserts that the 2 alleles for any given gene, or trait, are inherited. One from each parent. During gamete production, only one of the 2 alleles will be present in each ovum or sperm. Part 2. Chemically identical alleles are called homozygous. Homozygous refers to the condition in which a pair of alleles at a single locus on homologous chromosomes are the same. Chemically different alleles are called heterozygous. Heterozygous refers to the condition in which a pair of alleles at a single locus on homologous chromosomes are different. Sometimes alleles exhibit codominance, where neither chemically different version dominates the other. Codominance refers to 2 different alleles that are equally dominant. Both are fully expressed in a heterozygote's phenotype. Part 2. Chemically identical alleles are called homozygous. Homozygous refers to the condition in which a pair of alleles at a single locus on homologous chromosomes are the same. Chemically different alleles are called heterozygous. Heterozygous refers to the condition in which a pair of alleles at a single locus on homologous chromosomes are different. Sometimes alleles exhibit codominance, where neither chemically different version dominates the other. Codominance refers to two different alleles that are equally dominant. Both are fully expressed in a heterozygote's phenotype. And that's it for today. Thank you so much for watching my presentation. I love you guys and see you soon. Bye bye.